Hello everyone, today we will be discussing cell, the basic unit of life, which is a part of 8th class biology syllabus, and it's the second chapter in Telangana government state syllabus. First one I just wanted to skip, which is the basic one, what is science. So I wanted to start from cell, the basic unit of life. As you know, Earth is a very beautiful place where we can see different diverse organisms. They may be plants or animals, okay, and even in this plants and animals also, all the plants are of not same type, and even all the animals are also of not same type. And even if you see in rainy season, if water is stagnated for longer time in one place, then you can see some green color carpet. If you touch it, you can feel very smooth, which you feel like a velvet one. So that is nothing but mosses. And from the mosses, minute mosses, actually they are nothing but the small plants which are considered as algae, and from minute mosses to huge conifers. You can see in the cold places like Kashmir or the Himalayan range places, you can see all the cone shaped trees which are called as conifers. So from minute mosses to huge conifers, all these are the diverse plants. Now if you consider animals like from small insects, like a beautiful butterfly or a harmful mosquito or housefly, from there to a very big animal on the earth, an elephant, or aquatic organisms like a blue whale. So very big blue whale, all these have a very basic structure called cell. Even humans also have cells. But how could we come to know this? What led us to conclude this? All the living organisms are made up of cells. So to know about this, we should go 350 years back when the first microscope was invented, which laid the foundation to the discovery of the cell. So in this contribution, few scientists are named here in your textbooks. The most important scientists mentioned are Athanasius Kircher, Jan Swammerdam, and Antony von Leeuwenhoek, and also Robert Hooke. So these are the renowned scientists who contributed to the discovery of cell. Now, here you can see the box. Antony von Leeuwenhoek from 1632 to 1723, that is his time period. In 1674 was the first human to see living bodies like bacteria, yeast, protozoa, red blood cell, and the teeming life in a drop of water. He prepared several types of magnifying glasses and used these lenses to study about both living and non-living things using microscope. As I said, some of the scientists who contributed towards microbiology, amongst them Antony von Leeuwenhoek. He was the first person to see life in a drop of water like bacteria, yeast, protozoa, RBC, etc. Before him, the person who discovered cell was Robert Hooke. Let me tell you what actually Robert Hooke had done. Robert Hooke is a British scientist. He observed a piece of cork from oak tree. He observed a thin slice of cork under his self-made microscope. He was actually a fabric merchant. He used to observe the things with his self-made microscope. So that is how it happened to observe even a piece of cork from oak tree. Then he could find the honeycomb shaped structures which is seen in figure 2, and he called these as cells. Cell is a Latin word derived from cellula. Okay, cell is derived from cellula. It is a Latin word which means a little room. Actually, as he observed a cork, which is a dead tissue, so he could see all the empty spaces. But if you see, do you think that even the same will be with the living tissue? No, it will not be empty if you observe a living tissue. When Robert Hooke observed these empty spaces in the dead tissue, that is cork of oak tree, then he called those empty spaces as cells, which has the origin from Latin language word called cellula, which means an empty space. Later, in 1674, another scientist called Antony von Leeuwenhoek, he was the first person. He observed a drop of water from pond where he could see all the bacteria, and even he observed red blood cells, okay, blood from a mosquito. He observed many things, and he could observe some of them are moving, and he named them as animalcules. So first discovery of cell was done in a dead tissue. This is how it laid foundation to the discovery of the cell and also towards the development of microbiology. But now we are saying that every living organism is composed of cells. Then can we observe it in our daily life? Of course, we may not find oak trees here, but can we see the same type of tissue here? Yes, obviously, because generally trees will have the dead protective tissue, which we call it as generally bark, right? So if you just take a piece of matchstick, when you try to soak a matchstick in water for half an hour to 45 minutes, then it becomes soft. Now you take out the pieces of the soaked matchstick and put it under the magnifying lens, then you could see, or else you can see in the microscope. Do you know what mounting is? It is a part of making slide to be observed under the microscope. Generally we use simple microscope and compound microscope. Microscope is a device which is used to see the microscopic objects, that means which can't be seen with an unaided eye. In order to see them, we use the object called microscope, and in this we have a lens. Okay, with the help of this magnifying lens we will be able to see, and if we use only single lens, that is simple microscope, and if you use more than one, that is compound microscope. Generally in laboratory, in school laboratory, we will have both, but we use compound microscope where we can find both the lenses. Let me tell you a brief information about microscope. In compound microscope we have two lenses, where one is eyepiece lens where we put our eye, and another one is here we have objective lens. When Robert Hooke discovered honeycomb-like structures in a dead tissue called bark, he called them as cells, which is originated from Latin word cellula, that is nothing but little room. Even we can observe the same by taking a matchstick. You just take a matchstick and soak it for half an hour in water and take thin slices from it and place a thin piece of it on a slide with the help of a brush. You know the process how to make a slide. Just whatever you want to observe, you have to take the thin slices of it and you have to put on a rectangular glass piece, which we call it as a slide. Okay, all the thin pieces will be taken out with the help of a brush and will be placed here, and then we will place a round glass piece, which we call it as a cover slip, and we should very carefully place cover slip over the slide, because there is a chance of getting air bubbles. If we see these air bubbles under the lens, they will be seen very large, which we cannot see the actual one which we want to. So we should be very careful while putting a cover slip on the observable thing which is kept on the slide. So this process, without air bubbles, we have to put the cover slip on this slide, and this process is called as mounting. Okay, so after mounting, this slide is put on the stage. What is stage? Stage is nothing but a place where we put the slide to observe in a microscope. First, what a microscope is? Microscope is an instrument which is used to observe the minute things with the help of a magnifying lens. Again, this is of two types. One is simple microscope and other one is compound microscope. In simple microscope, only one lens is used, and if more than one lenses are used, that is compound microscope. That is given in annexure, that is in page number 25. See, this is the figure of compound microscope. You can see the stage. Stage is nothing but this is the place. Actually, here two clips are there. With the help of these two clips, we will place the slide here. Where we place the slide, that is called as stage or dais. And here, this 1, 2, 3, these are the revolving nose pieces. These are also lenses. And here, this is eyepiece lens, where we put our eye and we try to see through this. And this is the body tube. Here, this is coarse adjustment. With the help of this, we will try to adjust. That means what happens when we are revolving this, then the distance between revolving nose piece and this slide will be varying, so that we will make adjustments with the help of this knob, through which we can see that object clearly. This is one more thing. Reflector, that is nothing but a mirror. Light is focused here. When light is reflected through this mirror, things which are on the slide can be seen well. So this is the structure of compound microscope. So this is how we will do a slide which helps us to observe under a microscope. Whatever was observed by Robert Hooke, the same things which we can observe in a soaked matchstick also. If you see the structures like this, when you make a slide of matchstick and put under the microscope, then you could see all the structures like this, which was similar to the structures observed by Robert Hooke. So the discovery of cell by Robert Hooke was a milestone in the history of science. Now we have completed this dead cells, and as we said, the dead cells have empty inside them. And what about the cells which have life? Now let us try to do with the living cells by taking an onion. So all must be observed that how an onion will be. When you try to peel out the dried layers of onion, then you can see the fleshy succulent layers of onion. And even when you try to take out that outer peel, inside that outer peel you can see a very thin translucent membrane. Okay, when you try to break it, then you can see very clearly very thin translucent membrane that you can see. And you just take that carefully and put it in water, and with the help of a brush, again if you try to make a slide and put it under the microscope, then you can see the cells like this. Okay, actually the thin translucent membrane will be white in color. So in order to observe them very clearly, we use certain colors in the laboratory. Here we use safranin, which will be pink in color. Okay, let me remove all this. You can see, you observe the figure 3. See, in this it is without stain. So in order to observe them clearly, we use certain colors like safranin, which will be pink in color. So if you just put a drop of safranin, then you can see all these structure pink in color beautifully, like this only. Exactly, you can see the same figure what is given when you observe onion peel. Till now what we have seen, first whatever was observed by Robert Hooke, that is in a dead tissue, that is done. And even we can observe the same by taking a matchstick, that is done. And even we tested with the living cell, that is onion peel. And now what about the animal cells? Even animals too have cells, including humans. We said, let us try with human cheek cells. Can we observe the same? Sometimes we observe the white color thin layer in our hands when we brush in the early mornings. So here we are supposed to observe that only. In order to observe that and in order to take out that from our mouth, we have to make use of a toothpick or a wooden or plastic spoon very carefully. Without hurting yourself, you have to take it out. And again, after taking it out, put in water and with the help of a brush, put on the slide, and with the help of a cover slip, after doing mounting process, make the slide ready. When you put it under the microscope, you can see the structure like this, the same exactly which is shown in this. The same way you can find human cheek cell structure. So by this, what you can see, onion peel cells are rectangular, and even inside the human cheek cells here they are irregular and shapeless. So this is how we can see cells. This is in plants and this is in animals. What we learned today, discovery of cell, how the cell will be in plants, that is by taking onion peel, and next how the cells will be in animals, that is by observing human cheek cells. We'll meet in next session. Thank you.